Okay. Can I have the next slide? So the metal nanoparticles for drug delivery at the to target the cancer. So here, next slide. Right. So when you are going to talk about the cancer is it is somewhat different from the a normal tissue. You already know that. It is, for example, the first panel that gives a boundary line. The boundary line is nothing but the plasma membrane, and then lymph vessel, and then blood vessels. So these are the normal architecture of any tissue. So when you are going to see that the abnormal cell, they're supposed to change their their morphology. So when you're going to see the abnormal cell, that certain cells, they loses its own architecture, and then when the abnormal cells are going to be the abnormal cells started to multiply certain cells. So the changes that occurs in the blue color. So that cells are going to be modified their structure, and then it loses its own property to control the cell division process. So when the cell division process is going to be attacked, or it is going to be modified or altered at the genetic level. So what happens? Cells started to multiply in a faster rate, and it produces a mass of cells. So the mass of cells that will affect the blood circulation as well as in the lymphatic system. Can I have the next slide? Next slide. So here the different organs, because when you're going to see the cancer is going to affect a particular cell of a particular tissue, and by which it form a specific modifications in the physiology as well as biochemical reactions at molecular level. So that for example, here it is given as the breast cancer or cervical cancer, lung cancer. So based on the organ which is going to be affected, and the name is given as a breast carcinoma, cervical carcinoma, colorectal carcinoma, right? Next slide. So in what way the nanoparticle is going to support this research? In nowadays, each and every discipline, even if it is physics or chemistry or even medical sciences or genetic, the department of genetics, they are mainly looking for certain nanoparticle to support the drug delivery as well as to enhance the druggable activities. So here, sometimes some of the molecules that may be exist in the form of a polymeric nanoparticle or micelles or liposomes, or sometimes it's a nanogel. So, however, I would like to emphasize the importance of the nanogel. The nanogel which we developed in Harvard Medical School when I was doing my postdoc at the USA. So we developed a nanogel to prevent the post-operative cancer therapy. Post-operative cancer, because if once surgery is over, so what will happen? Some of the shredded cells, they may enter into the effect in human being. So we did that particular part of work, and we published that paper in the translational medicine. And then mesophorous nanoparticle, and then metallic nanoparticle, and then carbon nanotubes. So these are the various kinds of the nanoparticles which we use in development of drug or smart drug delivery, as well as for the treatment of various diseases, including cancer. Right? Next slide. So this is a molecule event that is taking place while nanocarrier is going to be used as a drug delivery agent. So when you're going to see that the cell surface is having the double membrane system. The double membrane system is having that receptors are going to recognize the drug of interest, and it will get internalized. If once the drug is get inside the cellular system, what happens? It is going to mix up with the cytoplasm, and it has a unique property to target two important cell organelles. One is mitochondria, another one is the endoplasmic reticulum. So once it is going to regulate the mitochondrial protein synthetic machinery, because it is an autonomous organelle, it can able to synthesize their own protein. So here the BCL2 family members. BCL2 family members are going to be expressed in the mitochondria. So the mitochondrial proteins that play a vital role in determination of the lifespan of the each and every cell. So here in cancer cell, this particular organelle, that is the mitochondria, is going to regulate the certain protein expression, and those proteins are essential for transporting the drug of interest into the nucleus. So if once it enters in the nucleus, it regulates the cell cycle process. So cell cycle checkpoints are going to be created. Cell cycle checkpoints are going to be created or generated through the nano drug delivery system. Next slide. Here, see, when you're going to use a specific drug for any particular disease, even for a diabetes or neurogenetic disorders or cancer, so frequently using drugs are supposed to develop drug resistance. So in order to overcome the drug resistant capacity, we are using the 4 different forms of nanoparticles. One is the liposomal nanoparticle, another one is polymeric nanoparticle, and third one is the inorganic nanoparticle, and then hybrid nanoparticle. So these nanoparticles are having the capability to carry drug of interest, or protein of interest, or nucleic acid of interest, and the nucleic acid based drug, that means the DNA vaccination or gene therapy, those things are going to be regulated. And then apart from that, recently in 2021, the Nobel Prize was given to Professor Karat, and for the development of a specific interaction which is made by immunogenic cells. Immune cells are having the capability to target the cancer. So particularly the TH cell, the TH cell, the T cell, T helper cell, T lymphocyte helper cell. The T lymphocyte helper cells are used to target the cancer. Next slide. Next slide. Sundar.
Sundara sir.
Next slide. Next slide. Yeah. This is a liposomal nanoparticle to overcome the cancer drug resistance. So here, the multimodal therapy is going to be adopted. Multimodal therapy means we are after the surgery. After the surgery, we are implementing the delivery of the cell of interest into the target site. Because when you're going to make a surgery on the tissue, if it is breast cancer or even if it is colorectal cancer, so when you're going to remove that in the tissue, so the blood vessels as well as the lymphatic systems are going to be opened. So through the lymphatic system, we are injecting immunogenic cells, and through these blood vessels, we are delivering the drug of interest. We are delivering the drug of interest. So here it is classified into monodrug or multimodal therapy or targeted therapy or co-delivery of a multiple chemotherapeutic agent and combinatorial therapy. So these are the various methods which was generated only because of the development of the nanoparticle based drug delivery system. Next slide. Sundar. Sundaraj.
Sir, please
Dr.
wait
Kannan, please wait.
for a minute,
Please
sir.
wait.
Sir, now it is ready, sir.
Still it is doing. தம்பி ஸ்லேட் தெரியலையே. Okay. So the another type is known as polymeric nanoparticles. The polymeric nanoparticles are polymeric prodrug, polymeric capsules, and polymeric dendrimer, and then nanogen. So these are the four different forms of the polymeric nanoparticles which are useful in biomedical industries. And it is also useful for the development of druggable prodrugs. So usually the recent days we are depending on the plants for targeting the cancer. So those phytochemicals are useful to deliver the drug of interest using the gold nanoparticle. One of my students, Dr. Meena, as well as Dr. Sujita, they did their wonderful job in modifying the nanoparticle using a specific drug that is known as doxorubicin nanoparticle. Okay, next slide. So here is the gold nanoparticle exists in different forms. So either it may be a nanosphere or nano cage or nano star or nano rod, or sometimes it may be a nano cluster, or sometimes it may be a nano porous material, and non-metallic substances like MSN and HMSN. There is a mesoporous silica nanoparticle, and another one is the halomesoporous silica nanoparticle, and then magnetic nanoparticles. These are the different forms of the nanoparticles that are existing. However, even if it is a single nanoparticle that is a gold, they are exist in 6 different forms. One is a nanosphere, nano cage, nano star, and nano rod, and last one is the nano clusters. So if it is in the exists the form of a cluster that can be able to accommodate more number of the drugs or phytochemical to target the diseased or cancerous tissue. Next slide. So this is the last type that is known as a hybrid type. The hybrid type is useful to target the biocompatible polymers and then phospholipids, and then the third one is the cell membrane. So the phospholipid is one among the major component of the cell membrane. So the cell membrane system is having certain receptors. They are exist in the form of ligand molecules. The ligand molecules are attached with the receptors, and by which the drug of interest is going to be delivered into the cellular system. Next slide. Next. Yeah, this is one of our research application. We develop a iron oxide nanoparticle. The iron oxide nanoparticle is useful to deliver the drug of interest to target the breast cancer. So this is the schematic representation. The schematic representation that reveals how we are going to synthesize the iron oxide nanoparticle, and then how it is going to attack the cancerous tissue. Next slide. Yeah, this is another publication from our lab. Dr. Santi. She did her work on delivery of doxorubicin. Doxorubicin is a breast cancer targeting component. So this compound is going to be attached with the palladium nanoparticle. So the palladium nanoparticle get pegylated. Polyethylene glycol is going to be added here, and then the polyethylene glycol carries the drug of interest, and it is useful to target the HeLa cells. HeLa cells, that is a cervical cancer. Next slide. This is from our lab. There is a new journal of chemistry. We published one paper in 2017. Dr. Bhanumati, she did her work on delivery of berberine using the silver nanoparticle. So the biogenic silver nanoparticle is used as a potential anti-cancer agent for breast cancer. The schematic representation that reveals how we are going to load the drug of interest, that is berberine, into silver nanoparticle, and after formulation of the silver nanoparticle, the particles are going to attach with the polymeric substances, and the BBR is going to deliver given in the form of a suspension solution. The suspension solution is going to attack only cancer cells, but the normal cells are always good. The normal tissue. So it won't cause any side effect in the liver, brain, lungs, and kidney. Next slide. Next. Yeah, this is recent publication from our lab. Dr. Vimala, actually right now she is working as a postdoc fellow in my lab for the past 6 years. She is working as a postdoctoral fellow. She developed a zinc oxide nanoparticle, but it is somewhat different when you compare with the previous literature. So this particular nanoparticle is having the capability to establish a photothermal targeted therapy for breast cancer. Photothermal targeted therapy for breast cancer. Next slide. This is a contribution from Dr. Ponraj. Right now he is working as a postdoc fellow in China. He developed a nanoparticle using the PVP, polyvinyl propylene. So the PVP is used to establish a titanium able to carry the drug of interest. The drug of interest is going to attack the cell signaling process through induction of apoptosis. Next slide. Again, there is another nanoparticle that is developed using silver nanoparticle. So the silver nanoparticle is specific to target the breast cancer cells. Next slide. So recently, Vivek, he developed a new strategy to target the cancer. That is a photothermal and photodynamic interaction created by nanoparticle that is known as carboplatin. So the carboplatin is having the potentiality to establish photothermal and photodynamic therapeutic agent. Next slide. So whatever may be the nanoparticle which we are developing. So ultimate aim is to target cancer. So in order to target cancer, So here, when you're going to see that how it is interacting or how it is going to deliver into the cellular system. So if once it reaches the cellular system, it is going to target the two important things. One is the nucleus, another one is mitochondria. So the mitochondrial membrane potential is going to be altered. So because of that induction, what will happen that it releases the apoptotic protein. So once the apoptotic protein is going to be released, it is going to interact with the nuclear material, and by which it induces apoptosis. Next slide. So in conclusion, I would like to emphasize highlighting the great bioavailability of the nanoparticle, and then after that, the bioavailable molecules are going to retain in the cellular system for a longer time, and it slow and steady releases the drug of interest to the target cells. So this is our conclusion. If anybody wants to clarify any doubt, they can raise their question. Now the session is open for discussion.